It's indeed a great pleasure for me, especially for me, to invite Dr. Rajendra Pilankatta. So, good afternoon all, and welcome to the last part of our webinar on viral disease. And it is a very pleasure in the sense Dr. Rajendra is a senior to me, and he is a former student of our department, School of Biosciences. When I joined here as a student, as a MS student in 1998, we were supported very much with our senior students, especially the students from the batch of Dr. Rajendra. And we were staying in the same hostel, University Hostel of Mahatma Gandhi University. And we used to visit our seniors' room. And the major purpose behind that visit was to learn how they are studying, to learn how the method, how they are utilizing to explore the subject, to learn the subject. And at that time, we were having only limited facility at the department itself. And at that time, we could see all our seniors, especially Rajendra, in his room with the textbook of Voigt, biochemistry, Voigt, Stryer, and Lenin, and Sesame. So that was the nature of culture, the academic culture of our university and the department at that time. And that, those experiences gave much excitement to us, motivation to us. And that are some of the very important reasons that we could do something and we could learn something and we could reach somewhere. So it's indeed a great opportunity for the juniors also to see the contribution of our bioscience in various areas. That is the main thing. Another thing is that Dr. Rajendra was a student of biochemistry, and his advanced specialization is in dengue virus and virology. So these are some of the things again that we have to learn how interdisciplinary the subject is. So with these words, I would like to introduce our respected guest today, Dr. Rajendra Pilankatta, and he is currently working as the head of Department of Biochemistry and Molecular Biology, and also lab in charge of COVID-19 RT-PCR testing lab of Central University of Kerala. And coming to his educational qualifications, he has completed his BSc in Chemistry from Department of Chemistry, Government College, Kasaragod. And then he came to School of Biosciences for his MSc in Biochemistry. He completed by 1999. And he has completed his PhD in Biochemistry from Department of Biochemistry, Osmania University in 2006. And currently he is serving many positions at Central University, and he just completing his job at Central University, and he was also the director of research of Central University of Kerala. Then he's in charge of COVID-19 RT-PCR testing laboratory, then Department of Biochemistry and Molecular Biology, and then he was a member of the Executive Council of Central University of Kerala, and he is also a member expert committee for the establishment of Institute of Advanced Virology at Trivandrum for the educational students. So we are in the state of Kerala is just starting a virology institute, and Rajendra is one of the important scientists contributing much to the establishment of such virology lab in the state of Kerala. Then he is a visiting professor, visiting associate professor at Duke in US Medical School, Singapore. Then Dean School of Medicine. He was a former Dean School of Medicine and Public Health, Central University of Kerala. Then he is associate professor of Department of Health. Then visiting faculty, he was also previously a visiting faculty of Department of Microbiology and Immunology, Georgetown University, Washington in 2014 to 15. And also he was in 2014 to 15 was a C.V. Raman Fellow of UC Department of Microbiology and Immunology, Georgetown University, Washington. Then in 2011 to 16, he was assistant professor at the Department of Biochemistry at Central University. Then in 2010 to 11, he has completed his postdoctoral research at Department of Microbiology and Molecular Genetics, Harvard Medical School, Boston. Then he is having another postdoc experience also at California Pacific Medical Center Research Institute, San Francisco. And he's also having experience as a research associate in the recombinant gene product division of International Center for Genetic Engineering and Biotechnology, ICGB, New Delhi. And his major research areas include dengue virus replication and virus-host cell interaction, adenovirus-based gene delivery, antiviral drugs, P-type ATPase, and cancer drug resistance. He is having, to his credit, a large number of high-quality publications, and he is having about 40 publications in highly reputed journals, one book edited, and he has completed 7 projects, and he has completed projects from all the important agencies like DSC, SERB, UGC, DBT, DBT BIRAC, ASCST, and ICMR. And with these introductory words, I request our most respected resource person, Dr. Rajendra, to start the talk. Sir, please.
Hi, namaste. Hi, thank you, Dr. Radhakrishnan, for giving a very elaborate and wonderful introduction, along with the sharing the sweet memories of our old campus days. And also, I extend my warm good afternoon to Dr. Arun sir. He was senior to me when I was doing my master's there. And our common meeting point was in Milma booth. I remember those old golden days. And I'm great that still we are in touch, in one way or in another way. And dear students, a warm good afternoon to all of you. And indeed, it is a great pleasure to, to share some viewpoints with you people in this occasion. And first of all, I also thank for inviting me for this an important gathering. So today, I thought of sharing, let me share my slides first. Okay. I think my screen is visible now.
Yes, yes.
Okay, thank you. So, so it is very, very, interesting to speak about transdisciplinary and interdisciplinary research and all. Very, very interesting. And rightly pointed out by my friend, Dr. Radhakrishnan, that, biochemistry to virology. They said, still I believe I am a biochemist, basically. So how it is interlinked, actually, that is pretty interesting. If you go through little bit, I think historically, that is pretty interesting. In fact, the genomics department, genomics is actually evolved by a biochemist, Craig Venter. He was a hardcore biochemist, actually. I'm not comparing myself to that, very eminent scientist, but I'm telling regarding the field. Anyway, today my topic, what I would like to discuss is new horizons of adenoviral vector-based gene delivery in the COVID-19 pandemic era. I know that this seminar has started 3 days back. So by now, all of you are knowing about the COVID-19 and its, after effects and, transcending COVID era now, actually. We are trying to transcend the COVID-19 pandemic era. So, coming back again, the historical perspective, 400 or 500 years back when we see, historically, we were haunted by the infectious disease, starting from smallpox. If you, go back with the smallpox, you can see that the ending of Aztec civilization onwards, everything actually, there is a huge connection with the infectious disease and evolution of human culture, living style, and, it's actually a selection pressure for human being when you see in a longer perspective, actually. So smallpox has dramatically changed, actually it helped us to discover a lot about vaccines. And similarly, plague was haunting us. I mean, still plague is in and around us. Sometimes it's, comes up. So what happened is, in fact, if you see the history in the last century, mid of the last century, till that point, we were thinking, okay, all, plague and all smallpox, everything went off, and, we have very good water, we have very good sanitization protocol, we have very good house, and we have done industrialization, we have developed a lot. Human race actually thought that, but actually now mid of the last century onwards, new viruses, when old things went to behind the screen, new viruses, infectious viruses and other bacteria and basically infectious disease, re-emerge. That's why emerging and re-emerging infections are happening on and off. So when you see in the second half of last century, you can see that, we heard a lot of thing about, dengue, chikungunya, so, and Chandipura virus, Kaisnur disease, forest disease virus, Ebola, like that, H1N1, H5N1, a lot of things are coming up and down, up and down. Human race is trying to adapt, actually. So if you see our own country during that time, I mean, why I'm telling you is some historical perspective actually gives us some confidence. History programs us for the future. That's the main thing. Even when we learn something about DNA structure, it's always, I tell the class also, it's better to see the history behind that. So history programs us, history makes us to think innovatively also. So what I would like to say here is, if you see the last half century post-independent country of our great India, our main problem was nothing else except the food security. That's why we were successfully evolved high green revolution, and then it didn't stop. Then we went to white revolution. Then we went to IT revolution. We are actually running parallel with the global scenario, but we also had a lot of attacked by this kind of infectious disease and many other natural selection pressure also. And why I'm saying again is, when you see the beginning of this current century, this started with the corona word, actually 2001 and 2002. That SARS-CoV in the China, from the civet cat origin. People, actually it killed around 800 people or something, So then again, MERS virus in 2012, actually, camel, origin, and then, it also killed a couple of, hundreds. And then exactly after a decade, nearly a decade, we are now, hello?
Rajendra, slides are moving, or it seems.
Yeah. No, I'm just going to, continue. Okay. It's moving now, I think, right? Can you see? Moving, right?
Yes, of course.
Okay, fine. So it is like that we were evolving and evolving further. But what I thought is today in this, important webinar, I thought, let me share some of the one important tool which was evolved during these days. That is none other than the, gene delivery methods. Gene delivery methods became an important tool in basic research and in, and also in the clinical research and, gene therapy and vaccine delivery. So many other areas, gene delivery methods took an important role, actually. So this gene delivery method was evolving with the novel, innovative platforms, now and then. So if you broadly classify gene delivery methods, you can have one is non-viral method and viral method. So non-viral method in the vaccine itself, in COVID-19 vaccine time, we have listened that, Moderna vaccine, that is the mRNA vaccine, and that is actually, lipid nanoparticle, liposome mediated, transfection of the viral genome. In mRNA, we are transfecting there, and there is no viruses involved, that just mRNA we are using. And also there are a couple of other physical and chemical methods, non-viral methods are available. And I'm not going in detail in those aspects, Today, I would like to talk about the viral vector-based gene delivery. So why that is so important? Because in the COVID-19 pandemic era, the one important thing everybody was talking about the vaccine, and we were successfully able to develop a vaccine in both the ways, non-viral method and viral method, viral vector-mediated gene delivery method. So if you see, there are so many different kind of viruses which we are seeing around, it's being manipulated for the delivery of gene. Why we have to deliver the gene into a cell? To either to modify the cell, genetically modify the cell, and also to express our gene of interest, our protein of interest inside the cell. So now there are a couple of examples, like the first one is adenoviruses. That's the one I am going to talk in detail, and some of the work what I am going to do in our lab also in this area. And then adenovirus will talk in detail later. And retroviruses. Retroviruses, you know that it's an what is that? It's an integrating virus where RNA virus, which gets the famous reverse central dogma was discovered in this virus. And retrovirus, only difficult part is it cannot infect the non-dividing cells. So, but still people are using this one for gene therapy trials and other genetic gene delivery platforms. Lentivirus has one more advantage, is that it can infect non-dividing cells also. So that is the benefit of both are coming, and both are having both are RNA virus which is going to integrate into the host chromosome. And then comes the pox viruses. Pox viruses is one of the the platform which is extensively studied, starting from the smallpox vaccine derived, vaccinia virus, vector, that also people are using extensively in this area. And another one is adeno-associated virus. See, adeno-associated viruses are called dependovirus. It doesn't replicate by itself. It needs either some of the viruses, virus factors to replicate effectively. That's what it is called, adeno-associated virus. In order for replication, it needs adenovirus factors, some of the factors derived from the adenovirus. That's why it's called a dependovirus. It's a single-stranded DNA virus. One thing I can tell you, that if there are so many students, I think in the master students here, if you browse through adeno-associated viruses and gene therapy, there are a lot of literature which is doing innovative work in that area, because the benefit of adeno-associated virus is it is an integrating single-stranded DNA virus into the host chromosome, but the location is very, very specific. It doesn't cause insertional inactivation in the host chromosome. But lentivirus and pox virus is a possibility of insertional inactivation, so it is a random integration. So adeno-associated vector is relatively very safer, but only thing is the construction of that and the propagation of this little bit tough, and it is less tighter, it is growing. And another thing is baculoviruses. Baculovirus is actually insect virus. This also being used for the delivery of gene to the target cells. And then comes the herpes simplex viruses. Herpes simplex viruses, its a genome size is huge, and more than 100 kb, and that actually this also being used, and the benefit of this virus is it can stay as in latent phase. But one thing I would like to stress upon here is all these viral vector based gene delivery, what we are doing is we are deleting the essential genes of the virus, or essential factors of the virus, genetic elements of the virus, so that it cannot replicate after the injection into the host system. And it is called replication deficient viruses. So instead of that region will be stuffed with our gene of interest. Let us see the next slide. This is how an adenovirus vector as a gene delivery vector works. Actually, you can see this is a adenovirus, and its genome is here. It is usually 30 kb size, and it's a double-stranded DNA, not circular, linear one. And here you can insert your cargo genes, gene of interest here. And this modified DNA can be packaged back to the viral capsid. That's what by genetic engineering. And this virus, when it infects to the cell, and it delivers its genome, directly to the nuclear pore. And you can see this, after injecting to the nuclear pore, through the nuclear pore, the DNA with our gene of interest enters into the cell and codes for the mRNA, and that mRNA again, results in the production of proteins, which is necessary for, it may be antigen, it may be some other enzymes, which is we are, actually we are targeting for the, which is actually our target or therapeutic target. Now, a little bit more about human adenovirus, genetic grouping. And the genetic grouping, if you see, adenovirus is the one which is extensively studied. There is also a lot of historical reason is there. Next slide, I will tell a few of those things. And if you see, human adenovirus in the genera, there are 5 different genera are there. You can see adenovirus in human beings, and you can see in reptiles, you can see in the frog, there are fish and so many other, species. It has been discovered, adenovirus, but human adenovirus is quite well characterized for various reasons. One of the main reason is for developing the gene therapy vector. That is one of the main reason. And also to understand the basic molecular biology. Many of the basic molecular biology has been discovered by the research work, hardcore research work on adenoviruses. So it is actually, 90 to 100 nanometer size virus, which is non-enveloped virus. It doesn't have any lipid envelope like a SARS coronavirus, where we are seeing a lipid envelope and a crown, spike. It is not there, and it is a non-envelope and icosahedral shape and double-stranded DNA it has. And if you see, there are 57 different serotypes of human adenovirus which has been characterized, and it spans across the 7 species, A to G. You can see, A, starting from A, it is G till G, it is there. There are 57 different accepted serotypes are there, and the infection mostly doesn't cause serious harm, issues in the clinical manifestation in the human beings. And if lower respiratory tract infection caused by serotype 7 and other things causes a little bit more complications. Otherwise, most of them are upper respiratory tract infection, causes the upper respiratory tract infections, and is not lethal basically. And serotype 5 is the one which is extensively studied for the various purpose. Serotype 5. This is the same serotype in which I am also working in my current lab here. And little bit more about adenovirus. It is better to understand some of the genetic organization, genomic organizations of the adenovirus genome. It's a long double-stranded DNA where there is left ITR and right ITR is there. Their packaging and encapsulation signals are there. And one thing you please focus here, there is a even A, even B, E2B, E2A, E4, and E. E stands for early genes. Whenever any virus, infects a cell, there are early genes which actually makes the preparatory tools for the effective replication and packaging of the virus, for the successful replication of the virus. Here, in the case of adenoviruses, even A is the immediate early genes. The moment virus enters, it expresses this, and this codes for many components, which is one of the important component is it pushes the cell towards the S phase of the cell cycle. S phase actually has been did lot of research work based on even a region. That's what I was telling in a molecular biology evolution time in 70s. These were one of the hot area, these areas. Cancer, cell biology, and replication, transcription. This was a hot area in 70s, actually, especially in MIT. And so what I would like to say is even A region is which pushes the cell towards the S phase, and even B actually inhibits the apoptosis of the cells. So basically, any virus, when it infects any cell, cell dies, virus is not going to get benefit. So that's what it stops the apoptosis of the cells. Even B actually inhibits the apoptosis of the cells. And same way E2, E2 region actually produces certain polymerases, and E3 actually E3 also shuts off host immune response or innate immune pathway. It blocks interferon response, mainly it blocks. And E4 actually, helps in the transport of mRNA from in and out of the nucleus to the cytoplasm. From the nucleus to the cytoplasm. So like that, so many late proteins are structural proteins. This is the final structure of the adenovirus, where you can see that a fiber and fiber axis is there, a knob is there, and hexane proteins are spread across. This hexane protein makes actually the capsid of this, and then you have a pentan base, and this fiber, and this virus is very, very stable. I mean, stable in the sense, in 4 to 5 degree, if you keep in the refrigerator itself, it's sufficient. See, I would like to stress one important point here is the whole Covishield vaccine. Actually, Covishield vaccine vector is adenovirus vector. That is, but it is not derived from human adenovirus. It is derived from chimpanzee adenovirus. It's a closely related non-human, primate. I mean, it's a primate. So chimpanzee adenovirus, they have derived. There is a reason for that. I will tell you that later. The main reason how we can effectively transport this vaccine across the developing countries with a small ice pack or small, 4 to 5 degree refrigerator, the reason was the stability of this virus. If it was an enveloped virus, we could not have done that. It needs, deep freezer and other stuff. So anyway, this is the electron micrograph of the Ad5 adenovirus serotype 5 virion. You can see it's 90 to 100 nanometer size. And this photograph, why I put specifically, because this is an historical development. You can see a red line here. And, this is actually called R-loop analysis, and using the EM radio labeling. This is autoradiograph actually. So then, what you can see that a small extension here. So this is one of the breakthrough work in 70s by Philip Sharp. Philip Sharp, who got Nobel Prize in 93 for RNA splicing. So basically, splicing of a eukaryotic RNA is discovered in the adenovirus model. That is the interesting thing. Okay, so they thought, what is this hanging? When they did the hybridization, they found an extra element hanging here towards the 5 prime, and they thought what it is. Later, also they compared the hexane coding mRNA in the infected cell in the cytoplasm versus corresponding DNA sequence present in the nucleus. What they found is something is missing, something is longer, like that there is a mismatch. Later on, the same question led to the discovery of RNA splicing by Philip Sharp, which was recognized by Nobel Prize in the 93. Actually, Nobel Prize was given for Philip Sharp, and Philip Sharp was a postdoc of James Watson. Philip Sharp, and they're all working in MIT, where they were working on the common target of molecular replication, transcription, and the institute was working on cancer biology. Now you could understand what your Benjamin Levin, your cell biology textbook, CARP, what it tells actually. Those histories are depicted in that book actually. What they were doing at that time, those work, and this is the outcome of the work. The splicing, RNA splicing was discovered in this. Okay, so now directly I'll go to the entry of the adenovirus into the non-immune cells. The why I wrote non-immune cells is immune cells actually is quite reluctant to receive the virus, adenovirus. There is no receptor for immune cells. That is one of the important thing I would like to talk towards the end of my end of my session. So it is a non-immune cells, like it may be fibroblasts, it may be some other epithelial cells. All these cells, it has a receptor for the adenovirus, that actually called CAR receptor, Coxsackie adenovirus receptor, and another co-receptors are the alpha-5 integrin. So what happens is virus actually binds here, and then the clathrin pit, through the clathrin pit, it actually as a vesicle enters into the cell. Within 15 minutes, less than 15 minutes, it will be in the endosome, where it is acidic pH. Acidic pH actually destroys its fiber, and then it starts to walk through the microtubule by the action of the dynein motor. Dynein, there is an ATPase driven mechanism. This virus actually moves through the microtubule and reaches the, within less than 90 minutes, it actually reaches to the nuclear core and ejects its DNA. And the moment DNA is here, it starts to synthesize its protein of interest. That is the very straightforward mechanism for the adenovirus uptake and multiplication. And you can see here, so absorption and penetration, bind to cell surface CAR receptor in the non-immune cells, and then transcription that I already mentioned, early transcription, late transcription, assembly, then it becomes an exit and cell lysis. So this is about the initial thing about the adenovirus. And there are, as any other adenovirus, this also causes production of cytokines and chemokines type 1 interferon. You can see that adenovirus, when it infects, and as I said, it goes to the endosome. Endosome, you have TLR7 and TLR9. TLR9 is called pattern recognition receptor, and which recognizes the viral genetic element, and then it actually induces the signal, and the signal finally leads to the activation of type 1 interferon. And also there are other pathways. Virus has another important element called viral associated RNA, VARNA, that actually triggers RIG1 pathway. RIG1 pathway also activates the production of the cytokine. And similarly, inflammasome. Inflammasome is actually caspase 1 dependent complex. Inflammasome also triggers the production of cytokines, which is pro-inflammatory. So these are all how anti-adenoviral immune response happens with the moment it enters. But what happens is virus actually shuts off all this pathway in the beginning when it replicates in the non-immune cells in the beginning. Okay. Now let us, these are the, what I was explaining is the basic things about the adenovirus. We should know what adenovirus is, because it's extensively used now for making different kind of vaccine platform and gene therapy platform. Now you see how we are making the virus in the lab. This is the virus isolated in our own lab here. You can see a clear, it's a density gradient separation of the virus. You can see a clear band of white band here. This is actually pure adenovirus, which we can actually pierce through this centrifuge tube and collect and do dialysis and use for your experiment. So this was actually how we are constructing in the lab. This virus is actually, you can clone the gene of interest, whichever you want. If you want the SARS-CoV-2 spike gene, you can clone that in the shuttle vector. In this, it's called a shuttle vector. You can insert the gene here and then do an homologous recombination with the PAD-EC1. PAD-EC1 is actually whole adenoviral vector. Adenoviral genome is here. Adenoviral genome in a plasmid format. And there is an homologous site here. So in the specific bacterial cell called BJ5183, we will co-electroporate and both will do the homologous recombination. And after that homologous recombination, what happens? This shuttle vector delivers its gene of interest, which is here, into the genome, viral genome. This is, then we will chop that with a particular enzyme, and then you will get a linear genome of adenovirus wherein you have gene of interest here. So in this vector, actually E1A is missing. In this vector, E1A region, which I explained in the previous slide, is missing, so that it cannot multiply. This is a whole virus, things are there, but it cannot multiply because it doesn't have E1A region. In that place, we have put gene of interest. We made some room there and then put our gene of interest or our cargo. Now, how to package this? When we are packaging, that is called a packaging cell. So we will put another, we transfect these cells into HEC293. So why this HEC293? HEC293 cells, which is the cell which is extensively used in research. HEC293 cells has been derived by transforming the cells using the sheared adenoviral DNA, actually, long back. Sheared adenoviral DNA, and 293rd passage, you got that actual stable cells, actually. So in a way, in that HEC293 cells, whenever you take any HEC293 cells, there is adenoviral sequence always expressing in that. In that you can find E1A region. So what happens when you put this one into the HEC293 cells, E1A region trans-complement, and this whole genome starts to get packaged as a full virus, and it is called a packaging cell. You will get the full virus package. Same strategy is being used for Covishield vaccine production, where you have a packaging cell, and instead of human adenovirus there, we are using what is that called chimpanzee adenovirus vector. So then once this virus is packaged, there is a couple of processes where you can purify and then do an ultracentrifuge, you will get the vector. And the interesting thing is, in this vector, this is now infective adenovirus. If you infect this one into any cell, it will, deliver the gene of interest which we have cloned, but it cannot infect normally into any monocytes or macrophages, immune cells. And interestingly, you can see there is a Scandi band here. I don't know if this you are able to see here this band in your slide there. So this Scandi band is actually a virus band where it doesn't have genetic material. It's a packaging deficient. Actually, only capsid is there here. It is like a virus-like particle. You can clearly see when you do. So because of the DNA, this is coming down and this is up, where it lacks the DNA inside that. And let me explain why I am telling all these things. First of all, why adenovirus is so important. One thing I told, Covishield vaccine and gene therapy platform and all. Why the one of the main aim, or one of the main benefit, or one of the main requirement when we do adenovirus vector for therapeutic purpose, we have to think of upscaling. We have to think of making it more safer and efficient. So all these characteristics are there for adenovirus vector. It is very safe, and it is insert capacity up to 7.5 kb genetic material you can insert in that as a cargo when you deliver into the target cells. And gene delivery is efficient actually. If you just put that a drop of the virus into the monolayer of the cell, it just infects. And also remember one thing, in the case of a vaccine and all, you have to inject onto the human being. In the case of Covishield, vaccine, it is injected intramuscular, and slow release is there. And there are so many animal experiments. What we are doing is by intravenous injection also. Virus efficiently enters and infects that. And it has a very broad host range and high expression levels. And you can get virus titer up to 10 to the power 10 or more than that. And it induces very good immune response and adaptive response. These are all the factors which actually made this vector as an important tool in vaccine as well as in the gene therapy trial, and also in the basic research. So if you see the history, again, literature, adenovirus vector application, gene therapy, one of the important, literature, that where Gelsinger, a boy, a patient was suffering from, cystic fibrosis, he was subjected to clinical trial actually, using one of the first gene therapy trial, and it was a failure actually. That patient died actually. They wanted to deliver OTC enzyme into that, but the patient died. The reason later on it came to know is they injected a very high titer of the virus, very high number of viral particle. For example, in Covishield vaccine, when you see the label, you can see it is written, there is 5 ml will be there in each vial, and there is actually 5 ml in that you are getting injection. Single dose is 0.5 ml. 0.5 ml contains 5 into 10 to the power 10 virus particles. That means 5 into 10 to the power 10 virus particle you are injecting per dose of COVID-19 vaccine. So same way, there in the Gelsinger experiment, with the clinical trial, they have used 10 to the power 13 viral particle into the body. That was one of the things, but it was a human adenovirus. Here we are using chimpanzee, but whatever it is, there high immune response, and that patient died actually. So a lot of setbacks were there in this area, lot of setbacks. And then after that failure, people, long time, they never looked into this area actually. Now, vaccine development also, then they later, HIV vaccine, they tried, it's a famous trial by, Merck company, and it was failed actually. Whoever got vaccinated, they got more HIV acquisition, because there comes some other reason, the pre-existing immunity and other reason. I'm going to explain that in next coming slides. And dengue vaccine, and also dengue and chikungunya vaccine, Ebola vaccine is one of the one. It's upcoming Ebola vaccine that also based on adenovirus serotype 5 actually. Lot of literatures are there, and oncolytic viruses. That is another important area. A huge amount of huge investment globally happens in this area, where tumor, we can kill the tumor cells using the virus, which is conditionally replicating in the tumor cell. It won't replicate in the normal cells. Conditionally, when it replicates in the tumor cell get lysed, and only specifically tumor cell will be dying and normal cell will be remaining intact. This literature, if you are interested, you can go through that. And then coming to my own work, during my postdoc time, this is the dengue vaccine vector, what we have used previously, where what we have done is, that dengue virus has 4 different serotypes, 1, 2, 3, 4. What we have done is we have collected, we have made a chimeric epitope by selecting E3 domain of the envelope protein of dengue 1, 2, 3, 4. E3 domain is actually, it has a neutralizing epitope, and E3 domain we combined from all 4 serotype, because in order to have a successful dengue vaccine, we need actually tetravalent vaccine, which should induce equipotent immune response. There should not be any skewness. So when you have a single vaccine, which should neutralize 1, 2, 3, 4 equally. If there is a skewness, that also is going to cause another problem. And instead of suppressing, neutralizing, it will enhance the infection. That's another thing I would like to talk after a few slides. So here you can see what ED3, envelope domain 3, carboxy terminus of the envelope protein domain 3 has been selected from all 4 serotype, and a chimeric construct was made, and that is a linker is glycine serine glycine linker, and it was then introduced into a viral vector, as I said earlier, and this vector was tested in the first in the cell culture platform, where we infected the HeLa cells with this tetravalent virus, which was also having a GFP tag on that. You can see that GFP tag tetravalent vaccine. That means when we inject this rat TG virus or recombinant adenovirus carrying tetravalent dengue antigen, if you inject that, it will express all 4 serotype antigen. So then what we did is we separately probed with the dengue 1, 2, 3, 4 antibody. Then we found all 4 antigens are expressing when you infect this one with the HeLa cells with the adenovirus, tetravalent adenovirus anchoring GFP. This is as a control. So it is clearly showing that it is expressing the protein. Then what we did is we injected into the mice. That is, this is the first dose, where we have used 5 × 10⁸ PFU particles. So that's just now I was telling, in a Covishield vaccine, you can see 5 × 10⁻10 particle we are injecting intramuscular. Here we have done it is an intravenous. When we injected, and then after 30 days we gave the first booster, and the 60 days the second booster. Then we have taken blood, from 3 different time points: the day 38 and day 68, and it is called homologous prime virus boost prime homologous regimen. That means the same vaccine will be injected for the boosting also. This you might have studied, that mixing of vaccine, when you might have learned, in the mixing of vaccine, when we Covishield vaccination, when we are talking about mixing of vaccine, mostly means heterologous regimen. That means first dose you are giving with the adenovirus, one particular serotype, and then you give with the another serotype adenovirus containing your gene of interest. It may be SARS spike or dengue spike, or you can give a plasmid directly, or you can give a nanoparticle containing the protein, specific protein in the second booster. So that is called heterologous. This is homologous. Mixing of vaccine means basically means this actually. So now After, then we took the, 3 different bleed from the animal, and then we tested for the dengue neutralization titer. The dengue neutralization titer you can see here. This is, we did homologous as well as heterologous also, same way. Okay, we did plasmid also, we injected. So you can see this brown color is indicating the homologous, other one is heterologous. Homologous gives better immune response. You can see first bleed, second bleed, and third bleed. After first bleed, you can see the enhancement of antibody titer. Here also you can see the enhancement of antibody titer. Third, there is no increase as compared to the second, but both are increased as compared to the first. Against 1, 2, 3, 4. All 4 serotypes are neutralizing, but if you see closely, you watch, it is not equally neutralizing. Here you can see dengue 3 weakly neutralizing, dengue 2 is highly neutralizing. So neutralizing titer is varying, it is not same. That's why we couldn't continue further that one. But the same thing is being, actually tried in another platform, Pichia pastoris, same epitope, that's going on. That work is going on in ICGB. And now coming back to same, see there, if you go through the clinical trial, sites, then to understand what are, then a huge number of clinical trials are going on in the against cancer, as well as, as a vaccine candidate also using the AD5. This is prostate cancer, metastatic pancreatic cancer, pancreatic adenocarcinoma, pancreatic cancer, and many other infectious disease. This is not the full list. If you are interested, I can share you that literature later on. It's a huge list is there where adenoviral vectors are used as a platform for gene delivery, for vaccination purpose, as well as for cancer treatment and other gene therapy purpose. And now let us divide into 2 era. Actually, till 2020, before COVID came, even though we had a lot of work, lot of clinical trial in practice, no AD5 based vaccine was in clinical practice. I mean, injecting thousands and millions of people injecting, that kind of a heavy practice was not there till COVID came. And once COVID-19 pandemic came, the whole world was running behind for vaccine. And at that time, then COVID-19 vaccine front runner became adenovirus vector platform. That is a chimpanzee derived adenovirus. Okay. Also, adenovirus serotype 5 derived vaccines, both were there. That is why I told you, it's a new horizon is opening up. I mean, these platforms can be managed effectively for many other pathogenic viruses. So basically, what I would like to is, it is not only new horizon for adenovirus vector, it is a new horizon for the people who are actually working and upcoming graduates, who are really enthusiastic to do things. A lot of things are there to do in this area, which is several things are unknown, and many questions need to be answered in this area. Why chimpanzee adenovirus vector we have used? The first reason is, as I said, human adenovirus is there everywhere. It's ubiquitous in nature. And before going to that, I will give you a brief thing of, it is updated by WHO in February 2021, where you can see AstraZeneca recombinant Shadox vaccine. That is what the same thing what we are using. And same thing, Serum Institute also finally came into picture, a Shadox adenoviral vector encoding spike protein antigen SARS-CoV-2 vaccine that we are using now. I don't need to explain much on that. And there are still other vectors platform like adenovirus serotype 26. This is a rare serotype as compared to the adenovirus serotype 5. This is actually Janssen company, and a single shot they are planning for this actually. Janssen company. Is adenovirus serotype 26. And then you see Sputnik virus. Sputnik was the another thing what we were using, listening in the news many times. Sputnik actually has an heterologous approach, what I was explaining just now, where the first dose is given by using the adenovirus 26 vector harboring SARS-CoV-2 spike. And second dose will be given by adenovirus serotype 5 harboring same spike protein. So why it is why differently we are giving? Because when you give the first shot one adenovirus, the same adenovirus when you give in the second shot, understand that adenovirus also mounts immune response. So when you inject the second dose, the pre-existing immunity against the viral vector itself can inhibit the immune response or boosting effect due to during the second dose. Okay, so that's why we are mixing first serotype and the another serotype. Same way, otherwise only single shot at 26. At 26, adenovirus serotype 26 is used as a single shot. Or, chimpanzee adenovirus, first dose, second dose we have given. We don't know about the third dose, whether it will be required. If it is required, can we give Shadox itself? So whether it will have enough neutralizing titer of antibody, whether it is capable to produce enough neutralizing titer, or how long the immunity conferred by this vaccine, Covishield vaccine, is going to exist. These are all the important questions here. Now you see adenovirus serotype 5, exactly the dengue vaccine platform, what I was explaining earlier, that work was done long back by us, and the same vector platform was used here, adenovirus serotype 5, and you can see that this is actually China vaccine, that is CanSino, but it was not used in many countries. And the problem here is adenovirus serotype 5 alone when you use, as I said, it is ubiquitously present in our neighborhood, in our surrounding. Our body already has antibody or cell-mediated immunity against the naturally occurring adenovirus serotype 5. So when you inject that as a vector, the vector may be eliminated because of this pre-existing immunity. That is the big question there. Now, towards this, we have done a survey in 2010 in AIMS, where in fact we published that work. And whether it is there, if so, India, there was no actually AD5 seroprevalence data till 2010 actually. It was not an, and for this purpose in India, what is the right, for that we have developed a assay platform, in-house assay in the lab, where we have used healthy human individual serum. We made a small selected sample, we published in Journal of Medical Virology, where we have done a serial dilution of human sera, and we combined with the adenovirus GFP, GFP expressing adenovirus, and a fixed MOI, multiplicity of infection of virus, fixed number of particles basically, and then mixed with the serially diluted sera. Then what we found is then infected into the HeLa cells, and then just collected the reading of the cells, GFP reading of the cells, and it was a methodology paper. What we found is there is a linearity when you increase the virus dose, but when you pre-incubate the different serum sample, if there is an antibody against the AD5, what will happen? Entry will be inhibited. So this reciprocal of this dilution was taken as a titer of the antibody in the human sera. So that way we found around 100% of the population what we have tested has AD5's antibody already existing. This work was done in 2010, and then you can see this is and with varying titer. 30% of the people, basically elderly people, have higher titer of anti-AD5 antibody in the human body. Okay, when you come to age-related correlation is there, and some other location and other things may be important. Especially tropical countries, we have high prevalence of adenovirus serotype antibody, but it doesn't cause any harm in the normal condition because it's simple mild febrile illness and it will just vanish. But when we use the vaccine, when you use a vaccine vector which is derived from adenovirus, this pre-existing immunity can counter that and it will blunt the immune response. There is a possibility. So you can see here, this is USA, it is only 30 to 60%, Europe 50 to 8, and Russia, Sputnik vaccine, when they release, Russia has totally different titer. It is not exactly like in India. So these antibody titer is one of the important player. I'm not telling it is totally blocking. And I would like to tell you one more thing is when they launched the CanSino vaccine as well as Shadow's vaccine also, even Covishield vaccine also, they have quoted this in our important paper. They cited, they told that this should be an important matter. We need to actually consider this also when we are, customizing the dose, or when you are going further with this trial. Of course, vaccine is very safe and effective, but there is skewness of immune response. In all individual, it is not causing the same level of immune response. So we need to see some of the betterment option for that. That's what I would like to say. And now coming to the another end of the same story, I told that antibody is preventing from the entry into the HeLa cells, but accidentally we have seen the same thing, same Ad5 GFP virus, when you pre-incubate with the human sera of with the antibody in a particular dilution, this adenovirus and antibody complex enters into the U937 cells. But if you put only virus, it doesn't enter because there is no receptor. When adenovirus is mixed with the antibody, it enters into the immune cells. That made us to think further why it is happening. Whether it is antibody dependent enhancement, that's the one we are talking in between. You might have heard somewhere. And which is exactly one of the important reason in several arboviruses, which causes, including dengue. This is one of the important problem where certain people, you can see dengue becomes very lethal and hemorrhagic fever happens. So it is proposed, there are two universities, two groups are there actually. Some people don't believe in this, some people actually they believe, but thing is there is something which need to be solved actually. So antibody dependent enhancement, whether it is playing a crucial, and interestingly we found the same thing in adenovirus when we mix with the U937 cells. But again, I would like to put a question mark here. The reason is, in real in vivo setting, the situation is different. You have a lot of serum factors and immune factors, complement, and so many other things, effector cells and cytokines. So many things are you have to take into, whether it will happen same inside the animal. That also we are trying to do now. And this is what antibody dependent enhancement of dengue replication happens in the case of dengue, where you can see dengue in the first, there are 4 serotypes. When first serotype infects, it gives immunity against that serotype, but that immunity is not able to cross protect the another serotype. So sequential infection of the heterotypic dengue virus, say one infected then becomes infected with the second, this cross reaction, non-neutralizing antibody, instead of stopping the infection, it actually makes the complex and enhances the uptake into the monocyte and increases the viral load. And when viral load is high, inflammation actually shoots up and causing the hemorrhagic fever and shock syndrome. Nowadays, it is called only severe dengue. Severe dengue is the new CDC term for that. And anyway, I'm not going to explain this much. Actually, time is running short. Otherwise, we could have gone in detail of this disease period and incubation regarding dengue. It is actually. So when what happens, when it's infects here and acute viremia happens very 6 to 7 days, and fever will be there. And then during this time, if any second infection happens, this may aggravate. That's what ADE. So the method is, a virus and antibody complex enters in a non-canonical pathway and not a classical pathway where opsonization happens and it gets everything destroyed. It is not like that. Virus actually evades from the immune complex and it actually multiplies and viral load shoots up and causing the cytokine storm. Cytokine storm is one of the reasons in the case of COVID also many people died actually because of the inflammatory cytokine. Now coming to some of the important research articles, Cell and other because antibody dependent enhancement of Ebola virus infection, and also and in the case of dengue, there are several literature in this area. Antibody dependent, Zika and also dengue Zika, because Zika and dengue are very closely related. I mean, in the sense both are flabby and the homology are there. Certain antibody may cross react. So whether Zika, if it is a subclinical infection where there is no asymptomatic infection, those people may have a possibility of enhanced infection. We don't know. Those all things we have to explore actually. We don't know. We need a lot of study, actually, scientific evidence rather than a lot of hypothesis. Hypothesis needs, but need to be verified, I'm telling you. Okay. So molecular mechanism of antibody-dependent enhancement, coronavirus entry, and feline coronavirus, which infects the cat and all, it is very clearly shown already that it is actually showing the same AD mechanism, you know. And also there was a Nature review article in the 2020 COVID-19 vaccine. When we design, we need to take into consideration of this. And I am sure that we didn't face any those kind of problem in the existing vaccine candidates, actually, you know. But always we need to understand the scientific principles, scientific theory, scientific and evidence, you know, how it works, and is there any similar situation scientifically. And towards this, we currently in our lab, we recently published one article in Analytical Biochemistry, where we have developed a assay platform, high throughput assay platform for ADE using adenovirus. The very, you know, logically, I told you that, you know, when a virus enters to the receptor, you can see the increasing fluorescence and it is a linearity. But when you make immune complex with the serially diluted virus, you know, serum, you are finding in a particular bell-shaped curve you will get. In a particular dilution, particular stoichiometry of virus and antibody, virus takes its path into the monocytes. So why it is happening? And what we did, we made a simple plate assay and an analytical platform for this. We, you know, when green fluorescence enhances, using that method, we can capture with a fluorescence spectrophotometer. So that platform also, this is a human sera what we have used. You can see here, this is a THP1 cells infected with the DFP, adenovirus harboring DFP. You can see 1 to 40 dilution of sera, 1 to 320 dilution of this particular sera. If you take another individual, it may be here. Another individual, it may be here. We don't know. There is a various antibody type. You can see a clear enhancement of the uptake of the virus at a particular dilution window. And then again, when you dilute the serum, that means when there is a low antibody titer, then it is not happening. It is like a zone of equivalence kind of thing. It is happening only a particular dilution. And we have, this is about human sera. We separately procured AD5 hexane antibody. Hexane I have already explained. Hexane antibody. Then we are seeing in a particular concentration dilution, 200 nanogram enhances the uptake. And this, you know, we have, what we have done is, when we deplete the serum, we are not seeing the fluorescence. Serum, when we deplete all IgG, we depleted using proteinase effros. Then there is no enhancement. So, and then we did, you know, different lysis procedure, and it's actually a methodology paper and semi-quantitative measurement of ADE. So actually it is a plate assay, 96-well plate assay what we have developed, because the reason is we actually looking, our main aim is, this is one of the platform for that. Main aim is to understand what are the host cellular factor which actually regulates the ADE or antibody dependent uptake of the virus into the cell. So what are the kinases involved? What are the lipids are involved? That we are actually screening now. And we got a pretty interesting screening result. You can see this. Actually, it is we are working on this further. You can see here, this is control. And there are, these are all kinase library inhibition screening study. Kinase library inhibition screening study, where we have used a FDA approved kinase inhibitor library. That is all drugs which is used for cancer therapy, like erlotinib and, you know, so many other drugs actually. So these drugs, you know, this library we are screening. When we screened, what we found is when you inhibit certain kinase, the uptake is tremendous. You see here, huge uptake is happening. When you use some inhibitor, kinase inhibitor, it is totally kills the uptake. And certain kinase inhibitor, actually you can see tremendously enhances. So there is some intracellular mechanism which is regulated by, as you know, how immune complex will enter into the cell through the Fc gamma receptor. Actually, we are trying to understand which Fc gamma receptor is responsible for this, whether CD16 or 32 or 64. And then we are trying to understand, which actually downregulates, which kinase is playing the role here while uptake. And one thing, these are all in the cellular platform. We have to try the same thing. Once we have some heat, definitely we have to try in the animal model. So the thing is, if you see, there are clear-cut modulators, kinase modulators, which is actually doing this particular important phenomenon. So that gives us a great hope to understand the ADE, and also, if some way we can minimize or better the vaccine vector, it will be a great help for the future. I mean, COVID, or we never know, as our history, I told in the beginning, history programs us for the future, so we never know. So we need to work. I mean, the COVID-19 actually gave us a lot of messages. One of the messages is to understand more virus and host interactions. What are the factors? What are the factors? Which are the type of viruses? Whether all, what is there any ubiquitous pathway? And what are the host factors? How we can manipulate it? Can it be a drug target? How we can better the existing vaccine platform? Can we have a better platform? For example, mRNA vaccine, it was used for the first time, actually, Moderna vaccine. So these are all the new avenues, especially for the upcoming graduates. I mean, it opens up a greater questions, and also to serve the society also through that way. And so I would like to summarize here, and I think I took a little bit more time, sorry for that. And basically, adenovirus vectored vaccines against COVID-19 opens up new horizon for developing vaccine against other emerging and re-emerging viral infection. That's what I was telling. And study on the interaction of adenovirus-based viral vector with immune cells will help us for a better specific gene delivery. And also, we developed in our lab, we developed recently a very ADE screening platform using a semi-quantification of GFP method. And we are actually exploring the different host cellular factor, which is actually influencing the uptake of the virus through immune complex into the cell. That may help us to understand better and to get more clues about the targeting this pathway for the therapeutic strategy. And before concluding, I would like to show this team. I would like to tell you one thing, that the moment COVID-19 started in 2020, in the beginning, Kasaragod is being a one area where there is not much medical facility. So it became our university was requested by the authority, can we do this? So our molecular virology lab, as a team, you can see a big team, research scholars, and my colleagues, Dr. Samir is there, and Dr. Rishiram Ramanan is there, and my research scholars are there, and all these people just came together. We made a good team, and we started as the 8th COVID-19 RT-PCR testing lab into March 2020, and approved by ICMR. Now also we are actually helping the sequencing of the sample. We have completed many samples, vaccine breakthrough and all, in collaboration with the IGIB New Delhi, and we have completed more than now it is actually still more, 2.2 lakhs COVID-19 RT-PCR test in our lab. So these are the team which is behind that, and it is a through which this we could do a great service for the society, our neighborhood. And I would like to acknowledge all my students, Ranjit, Ladika, or Reddy completed, Ranjit, Ladika, and they are about to submit, Arun Kumar also, and Anjali and Ashwati, recently joined. And these are my collaborators, Dr. Swaminathan, Naveen Khanna, Subhash Vasudevan, and Dr. Padmanabhan, and Dr. Manoj, and Dr. Unni, Dr. Swapna Nair. She is helping us in the nanoparticle-based work. We are working in it. We just funding agencies for us from various agencies, as Dr. Radhakrishnan mentioned in the beginning. And CUK, Kerala Biotechnology Commission, KSCST is constantly supporting us. DST and Department of Biotechnology also. And thank you all. This is our upcoming campus. Thank you very much for giving this great opportunity. Sorry, I took a little bit more time. I think I should have made less slides. Sorry for that. So thank you all. Thank you very much.
Thank you very much for your kind support to us. And now we have some questions from students in the chat box. Please.
Okay.
Maybe it's the first time we are getting a lot of questions just before the speaker finishes his talk.
So that means, I don't know whether it is understood or not understood.
Anyway, it's a very welcoming sign.
I am very happy, to get some question means. So, sir, according to you, which combination of vaccines give better immune response against COVID-19? Which combination of vaccines, in the sense, see, this is one thing what we are using in India now, Covishield vaccine. So I am sure that based on the existing literature, it would be better to go for heterologous immunization in the next dose, possibly. But I don't know, actually, if you see Oxford AstraZeneca, so there is one group called, you can, browse through, Professor Dan Barrow. Dan Barrow is in Harvard Medical School. In fact, I had a chance to, interact with him when I was in Harvard. He is one of the leading person who is doing vector development in the case of adenovirus. Who was the leading person in the HIV vaccine trial also? They developed a couple of chimpanzee-derived adenoviral vector, and later on they collaborated with the Oxford. And Oxford group, when they did the human, animal study, what they have used is vaccinia viral vector they used for the next dose, combination dose, along with the chimpanzee adenovirus vector. So I don't know whether they are going to use the same thing here, in this regimen. There are a lot of other, factors are there. They have to do clinical trial and they have to prove that, and DCGA should have approved. A lot of questions are there. So according to me, if you ask, heterologous will be a better option with the vaccinia system. Anything else in that regard? I think Aditya, Shashi.
Aditya, Shashi, is it clear to you?
She can unmute and speak.
Clear,
Okay.
sir.
Yeah. Okay. Any more question in that? Okay.
Yeah. There are some more questions are there.
Please, sir.
Miduna.
Sir,
Hello. Hi.
sir, can dengue antibodies provide immunity against COVID?
Not possible, because I don't think it will neutralize. It's totally different antibody, no? Dengue antibody is against dengue. It is targeting the normally envelope protein of dengue virus. COVID antibody mainly for this COVID spike protein. So antibody antigen interaction is very specific. So even if they show, and another thing, they are not belong to the same group or family also, far away. They are, dengue is a flavivirus actually. So it is not possible.
Amira.
Can
Yes, sir.
Please.
Sir, can you please elaborate on this oncolytic viruses, its scope and the future, all that?
Yeah. Oncolytic virus, there is a lot of scope is there, a lot of work is going on. If you want, I can share the literature. One of the review article is there. Just I was sharing that. The thing is, the basic principle is, as I mentioned you, adenovirus vector, when we create, we delete the even a region, for example. Delete even a region is essential for the multiplication of the virus. Okay. Now, if you infect the even a deleted virus into a tumor cell, Suppose tumor cell, for example, P53 is lower, less expressed in tumor cell, right? Tumor suppressor is not there, P53. So when usually P53 and even A there is an interaction, and P53 is lower, this adenovirus selectively and specifically will multiply in the tumor cells only. Are you getting my point? So then what will happen? The cell will lyse end of the day. So if there is a tumor here, and I am seeing so many cells are here in the tumor surface, and of course after certain area there will be normal cells. So adenovirus will infect the tumor cell as well as normal cell. But this adenovirus selectively multiply in the tumor cell and lyse the cell, and it kills the cells. And normal cells, it won't replicate, it won't kill the cells. That is what called conditionally replicating virus or oncolytic virus. There are various molecular methods to, do that. And, you can specific targeting into the virus, tumor cell or P53 expression based modulation. All these things are there. It is a big area actually, oncolytic virus. Basically, principally selectively multiplying in the tumor cells so that it will lyse the cells, other cells will be intact. And another thing, you can see there is a movie by Will Smith, I Am the Legend. Please see that movie. That is actually oncolytic virus experiment. They are explaining in that also. I Am the Legend. It's a good movie by Will Smith.
Okay, sir. I'll definitely watch that. Thank you.
Share the literature also. Thank you.
If there are other questions, please.
Dr. Rajendra,
Yes, sir.
in virology research, where we actually stand, I mean, India and Kerala, in terms of research and
You
developing new techniques.
comparing India versus Kerala.
Yeah.
I mean, national scenario where we are standing, right?
Yeah, of course, yeah.
What I would say is we need IAV-like centers more. I mean, I know that it is tough to establish, but thing is we need better facility first of all. Better facility, and we need to train more graduates in that area. We are lacking the manpower. That is one important thing as far as Kerala, I feel. Even our, state university, there are not many virology lab actually, or virology research groups. So, and also we need more training programs, especially in the microbiology department they can train. We need to give more trained graduates coming out in this particular area. That is one thing. And also we need a BSL-3 facility, and BSL-4 is not that easy. At least a very good BSL-3 facility is required to handle these high-end viruses. That's one thing. So we are lacking in terms of facility as well as trained and skilled manpower. Both are having a shortage here. And we need to attract global, the experts, and especially dynamic postdoctoral people and other people from outside to our universities through some special schemes like we have Ramanujan fellowships and Inspire faculty kind of things, Ramalinga Swami fellowship and all. We have to give more provision in our state universities, because Kerala is the state which need to give more stress to the infectious disease, especially viral infectious disease, because we are suffering from, one side Nipah and dengue and chikungunya, and this side if you come border areas, Kaisnor disease forest virus also is there in Karnataka soil. So needs our all state universities, we need more and more programs and training and skill development programs and other things. That's what I believe. And national scenario, if you see, there are some labs, CSIR labs, like CCMB, they have wonderful facility. IAC, couple of my friends, they are doing wonderful. NCPS also, NCPS not many groups are working. And the NIV lab is there, not DRDO labs are there, Gwalior and Delhi, 2-3 labs, National Institute of Immunology, ICGB, and the Translational Health Research Institute. All these people are working on that. There's a group actually. And unfortunately in Kerala, we don't have CSIR. We have only one CSIR institute is there. but I don't think there also we don't have more virology focused area there. Missed actually. But happy that Dr. Stalin is working in ISER. That is one group is there actually.
Yeah, right.
We need more groups in our university, state university, so that more graduates will come. They will start working on this area independently. That's what I believe.
Yeah, thank you.
Thank you, sir. It's a good question actually. It's a good question and expensive question.
Yeah, true. The money is one of the biggest problems
Everywhere.
for virology research.
Virology, that is one of them. And I have an ICMR project sanctioned. See, 1 year. Since 1 year I'm waiting. They are not releasing single penny. A small project. So a 30 lakhs project, ICMR, dengue project actually. But I didn't get that money.
Thank you, Dr. Rajendran, for sharing your valuable time. I know you are very busy. Even he is holding many charges of the Central University, even with he's like a virological research. He is also involved in many other responsibilities of the university. So I know your busy schedule. So thank you very much for supporting us, and we will be requesting your help always.
Sure. MD University means my Taravada.
Yeah.
Because എനിക്കേണ്ട ആവശ്യമില്ല അതൊക്കെ നമ്മളാക്കിയ തറവാടാണ്. See, one thing I think students